people were saying, oh, well, if it's set at a particular, you have a certain number in the readout, it should be functional. It should measure correctly. But remember, if you don't stop at the first stop, it does not measure correctly. You have to push this thing to the first stop and then let it up slow. If you push beyond the first stop, you don't know what you're measuring. So finesse is very important here. Make sure you use these things correctly.
He accidentally leaned in and went.
I did not.
So are you taking it tonight so that it's fresh, and that's why we're restreaking tonight?
We're restreaking. The other thing that sometimes happens, we have a primer set. If we try to run the 8-4 1530 primer set and it doesn't work, I can try 8-4 with 1492 and see if that works. So there's a couple of different things I can do. Okay, this is the laboratory that we're doing officially tonight, which is the mini screen, rapid isolation of plasmid DNA. There's some information in the laboratory manual about plasmids that probably what I should say is it's usually true of plasmid, but it's not always true. Just like bacterial chromosomes, most textbooks will tell you that the bacterial chromosome is a covalently closed loop. I think I've said that to you many times. It's covalently closed circular DNA, CCC DNA. They aren't always. Streptomyces have linear chromosomes. It wouldn't surprise me if Streptomyces have linear plasmid. But for the most part, plasmids are circular. Okay, so what are plasmids then? They're small extrachromosomal loops of DNA. Extrachromosomal means they're outside the chromosome. In case you wonder, that hula hoop that's sitting up there on the cupboard, that's a plasmid. It's a model of a plasmid. So it's got genes on it. It's got yellow genes and red genes and blue genes, and it's had some splicing done to it. That's why it's got tape on it. If you were to take that plasmid, that model, and go stand beside a quarter-mile track, the quarter-mile track would not be big enough to be a chromosome. But if you take that plasmid and go stand beside the cross-country track, how many of you know where that is? Maybe not. But it goes from behind the gym, it goes wandering way the heck out there, over by the freeway and around and comes back up. Then you have a pretty good size comparison. Plasmids are very small compared to the chromosome. So they are very small extrachromosomal loops. Here you see a representation of a plasmid, a single tiny little loop, and the chromosome itself looks more like a push ball. Got lots of DNA there. So there were some representative plasmids mentioned in the lecture, R-factor plasmids that carry genes for resistance, F-factor plasmids that carry genes associated with fertility, actually horizontal gene transfer. For the most part, plasmids are said to have genes that are not necessary for the cell. They don't absolutely have to happen. Yes? So exercise? 209. Sorry. It was at the beginning, but I went by it really pretty fast. The other thing about plasmids is typically they can reproduce independently of the chromosome. What does a sample of DNA have to have in order for replication to be started? You have to have an origin. The origin of replication, that's where replication begins. So if they're going to replicate separately from the chromosome, they have to have their own origin. Now, this plasmid replication you're going to watch here has a serious flaw. See if you can tell me what it is. Well, what's wrong with that? Did you notice those plasmids were coming up there one at a time? Yeah. DNA replication is always semi-conservative. 1 becomes 2. 2 become 4. 4 become 8. Okay? They were coming up there one at a time. That's not how it happened. But anyway, so they replicate. They have their own origin of replication. They're going to replicate typically much faster than the chromosome because they're so small. And as a consequence, oftentimes cells have more than one copy of a plasmid. they tend to have very large copies of plasmids. We have this E. coli model up here, in case you wonder what this box is about. So, in this particular sample, this is the chromosome, and this was supposedly measured so that it's an accurate representation of chromosome size. And someplace on this piece of yarn, there's some little black stripes that represent genes, little tiny segments. I don't know where they are, but anyway, this is the plasmid. So plasmids are little compared to the chromosome. They're much, much smaller. And again, they're going to replicate on their own, so they're going to tend to replicate much faster than the chromosome. Those other things that were in there fell apart, and they were too big anyway, so never mind. The number of plasmids of a particular type that are inside of a cell, that number is called the copy number. And again, it can be it's variable, but sometimes quite high, maybe 150 or 200. So there can be hundreds of these tiny little loops of DNA, these plasmids. If they have very high copy numbers, then they're often used as cloning vectors. What does cloning mean? Making copies. So you'll see that term used in a lot of different applications. If you do anything like Photoshop, you have a clone tool where you can copy sections of things. Cloning just means copying, but what's a vector? We talk about vectors, mosquitoes, ticks, fleas. So a vector is something that transmits a potential disease-causing agent. When you're talking about a cloning vector is basically a piece of DNA that can pick up DNA from some source and carry it into a cell and replicate it there. So plasmids can be cloning vectors. What that means is they can be manipulated. You can put DNA into plasmids, and then if you put those plasmids into cells, because they have their own origin of replication, they'll reproduce inside the cell. And they'll reproduce whatever you give them, whatever they happen to be carrying. Typically, to be a success as a cloning vector, these plasmids have to have some characteristics. One, they typically have to have high copy numbers, so that they're going to replicate themselves multiple times over, so you get lots of copies. The other thing they have to have are little sections of DNA called marker genes. Marker genes allow cells that are carrying those plasmids to be recognized and often allow them to be selected for. So we have some plasmids we're working with tonight. They're called PGEM, PGLOW, and PUC19. They're all carrying a marker gene, and the marker gene is a gene that encodes the enzyme beta-lactamase. We talked about beta-lactamase in lecture. Beta-lactamase is an enzyme that breaks down beta-lactam antibiotics. So how would you select for bacteria that are carrying that plasmid? Think about selective media. You want to select for bacteria that are carrying the beta-lactamase enzyme. You want to separate them from bacteria that aren't. What do you put them on? Beta-lactamase. You put them on medium that carries some kind of beta-lactamase antibiotic, any penicillin. We're going to be using ampicillin. So if you grow them on a medium that has ampicillin, then only the cells that have plasmids are going to be able to survive, because the plasmids are carrying that beta-lactamase enzyme. So this is, again, typical of these the cloning vectors. They have these marker genes. The other thing that they tend to have are multiple cloning sites. They have these because they have been genetically modified. So these are engineered plasmids that we're working with. And cloning sites are sites where restriction enzymes can cut the plasmids, and then that's where you can insert a piece of DNA that you want to copy. You can take DNA from anywhere and put it into a plasmid. The plasmid will carry it into a cell and replicate it. There's some little kinks to this system, and I'll explain that in just a moment. But this is an example of a plasmid. This diagram is made very large. That is, the cloning sites are made very large so that you can see them. So here's our plasmid, and up at the top you see the letter sequence GAATTC. Notice it's on one strand reading G to C from left to right. And then if you look at the sequence on the inside ring, it's GAATTC going the other way. That is a true palindrome, a sequence that reads the same forward and back. So those palindromes are often cut sites for restriction enzymes. In this case, the restriction enzyme that's being used there is called EcoR1, and EcoR1 cuts between guanine and adenine. So you can see the cut site between the G and the A up there, and then between the G and the A over here. If you subject a plasmid to that particular enzyme, EcoR1, it will always cut at that location, and it will take what was a loop, closed loop, and open it up. If it's open, then you can take any gene of interest that has been cut with the same enzyme. It'll have complementary ends that can hydrogen bond, and that way you can stick your gene of interest into the plasmid. So that's what's been done here. The purple part is some gene. It could come from anywhere. It's just called DNA to be inserted or gene of interest. Here it is inserted, and AATT binds to TTAA. Hydrogen bonding between these initially. But if we want to stick that in there, what's the enzyme that will stick segments of DNA together? That is, form phosphodiester bonds between DNA segments. Ligase, our glue enzyme. So you use ligase, and ligase will build a phosphodiester bond and stick this thing in. And then whatever that is, that gene of interest is going to be replicated right along with the plasmid. So again, these plasmids that are built, they've been genetically modified so that they'll have a whole series of restriction sites. You can cut them with different restriction enzymes. Different restriction enzymes have different sequences that they look for, and they typically cut somewhere within that sequence. Basically then what cloning vectors can do, they can take DNA in, you can put DNA into them, they'll carry that DNA into a cell and then they'll replicate. Because they have their own origin, they'll replicate it many, many times over. This is how DNA was replicated before the PCR was developed. Before the polymerase chain reaction allowed for replication of DNA in vitro, this was replication of DNA in vivo. So people were replicating DNA inside cells, get the cells to do it. And of course, bacteria grow really rapidly, so if the bacteria are carrying multiple plasmids, you can get lots and lots of DNA very quickly. Again, this DNA can be from any source. However, if you take genes from eukaryotic organisms and you stick them into prokaryotic organisms, such as Escherichia coli, can E. coli translate? Can it transcribe and translate genetic material from a eukaryotic cell? Let's just say we've got one gene from a eukaryotic cell. If you just take that gene and stick it into E. coli, are they going to be able to deal with it? The answer is no, because eukaryotic cells engage in post-transcriptional modification. Eukaryotic chromosomes have introns, and introns have to be taken out, and prokaryotic cells don't have spliceosomes. They don't have the small nuclear RNA. They can't take out introns and put together exons. So if you want to take DNA from a eukaryotic cell, what you have to do is go into the eukaryotic cell and find the messenger RNA that encodes the protein you want. For example, if you want to take enzymes from pancreas, let's say you want human insulin, you can't just take the DNA that encodes human insulin. Instead, what you have to do is go into the pancreas and you have to get messenger RNA after post-transcriptional modification. It's handy that that modified RNA, remember, introns are taken out, exons are hooked together, and then there's a cap and a polyadenine tail. So that a processed messenger RNA, a messenger RNA that's ready to be used for translation, has a polyadenine tail on it. And so what was developed were these columns of, they put glass beads in these columns, it was basically like tubes, but they had poly-T sequences stuck to the glass. And they could dump the RNA in there, and it can be mixed with a whole bunch of stuff, but the messenger RNA, the poly-A tails would stick to the poly-Ts, and they'd catch them. And then they could, and so that was, you had to get messenger RNA post-transcriptional modification. And then you have to reverse transcribe that. What do you think reverse transcription is? We know what transcription is, right? Taking the information that's in DNA, making RNA. You do reverse transcription, you take that messenger RNA and you reverse transcribe it into DNA, that's the DNA now that you can put into the prokaryotic cell, because the introns are gone. Tricky, no? There's another little problem that comes up, and I'll talk about that after a bit. Actually, it has to do with restriction enzymes, but later. Okay, so cloning. Cloning is a little bit like the PCR, but again, PCR happens in vitro, it's in a tube, and cloning happens inside the cell. What's a serious advantage that cells have over that little PCR tube? Let's say that you want to clone the gene for human insulin, and you've gone through this whole process of getting the messenger RNA that encodes that gene, and now you've got lots of copies of that gene. Why did you take it in the first place? Because you wanted human insulin. You can copy DNA using the PCR. You can't express it, and cells can. So if you take the gene for human insulin, go through this modification business of catching the messenger RNA and reverse transcribing into DNA, you put that DNA into E. coli. E. coli can not only copy the DNA many times over, E. coli can translate, and they will actually crank out human insulin, and they do. So there are now commercially available human insulin that is used to treat diabetics, and it's produced by Escherichia coli, which is pretty tricky. Okay, all of this is part of what's called recombinant DNA technology. Here's our plasmid reproducing, and it's got the gene of interest in it, and it's still reproducing all wrong, one at a time. I should fix that. Well, okay, tonight we're working with a plasmid called pGlow, and pGlow is carrying a gene from a jellyfish. The jellyfish is Aporia victoria. The protein is called green fluorescent protein. And so what we're going to do tonight is we're going to extract the plasmids. We're going to extract 3 plasmids, pGlow, pGem, and PUC19. And then later, we're going to put these plasmids back into E. coli. And when you put the green fluorescent protein into E. coli, they will express it under certain circumstances, and we're going to get E. coli that are fluorescent green. That's kind of fun. So, for this exercise, you're going to be working in groups. If you're at a table, and most of you are, where you have a table with 6 people, if there's anybody that wants to volunteer to be the 6th person for the back table, then we have, well, somewhere we're going to have a group of 2 people, so it doesn't really matter. It can be the back table, it can be the side table. Right now, it's the back table. So you're working in groups, and each group is going to extract all 3 plasmids. There's actually an advantage to having 2 people in your group, because this is another of those exercises where you get points if you're successful. You get points for the PCR. Now you might ask, have I given you points yet? The answer is no, because I don't know if your PCR worked. We'll find out when you run your gels. We'll also find out when we run gels if this plasmid extraction works. Which reminds me, we're supposed to be running gels on Tuesday of next week, which is Halloween. How many people are not going to be here because of Halloween? So, because you get points for the plasmid extraction and you get points for the gel, I'm thinking maybe I should flip-flop those labs and do gel electrophoresis the lab after. That way, people that have to run away for Halloween, because after all, it's Halloween, we won't be doing anything that requires points on that night. Actually, there is something that requires points, but you can do it at home. It's fine. I think that's what I'm going to do. So tonight we are going to extract plasmids, but we won't run the gels on Tuesday. Instead, we'll do restriction enzyme cutting of DNA. So, if you're a group of 3 people, in that group, 3 plasmids are going to be extracted. So, PUC19, pGem, pGlow, and we're going to have these tubes that we're going to be using. We can see the set buffer tubes that are displayed up on the screen. Notice they're different colors. It's really difficult to put a lot of labeling on these little tiny Eppendorf tubes. So instead, our plasmids are going to be color-coded. These colors are consistent from semester to semester. So the blue tubes are used for the plasmid PUC19. The orange tubes are for the plasmid PGEM. And the green tubes are for the plasmid PGLOW. It makes sense for PGLOW because PGLOW is carrying the green fluorescent protein gene, so it should be green. PUC, well, University of California colors are blue and gold, so I guess the blue makes sense. I don't know what gems are orange. I'm trying to figure that one out, but it doesn't matter. So a group of 3 people is going to get 3 different colored tubes. If you are a group of 2 people, you still need 3 different colored tubes. So then we're going to have these plates, and each plate has a lawn of E. coli, and the E. coli are carrying different plasmids. So this plate shows you a lawn of E. coli DH5 alpha that's carrying the plasmid PGEM. These plates are all labeled TSA AMP at the top, which tells you there's ampicillin in there. Again, the ampicillin is to help the cells recognize they have to keep making their plasmids because otherwise they can't grow on that. By the way, if you ever see these plates that are in the refrigerator that say TSA, actually the label will say TSA AMP, don't assume it's just TSA because it isn't. It's TSA with ampicillin in it. If your bacteria don't make beta-lactamase, they can't grow on that. This is a lawn culture, so you don't see much on there, but the bacteria are all over that plate. So it's kind of hazy patterning in here. They're all over that thing. And that's what the plates are going to look like. Your job is to very carefully scrape the cells from 1/4 of the plate. 1/4. So this is telling us the different strains. The strain that's called JM83 carries PUC19. The strain called DH5 alpha carries PGEM and PGLOW. Please pay attention to the labels and don't mix these guys up. This plate has been cut. It's not necessary to do that. But notice that now the lawn has been removed from 1/4 of the plate. Don't use less than that. So don't just take a couple of swipes and consider that to be good. You need all the cells off of the 1/4 of the plate. Don't dig into the auger. You're just going to get cells. If you have one of those loops that's made with a double wire that becomes a single wire in the loop, don't use it. Those things don't work. For this exercise, you can't use those things. So if you have that kind of loop, see me about getting a replacement. Those things are worthless. Yeah, it should be the same wire that makes the loop. Oh, okay, yeah, like that's the right number. Yeah, that's fine. If your loops are really small, you might try either modifying them or getting one that's a little bit bigger so that it doesn't take you so long to get the cells off. The question is, what are you going to do with the cells? You're going to put them into the set buffer. So
there's always so many of them. No. Surprised.
So you have these tubes up here. These tubes have 150 microliters of set buffers. And that's not very much, so there's a little bit of liquid at the bottom. And what you're supposed to do is get your cells to get off the loop and get in here. You do not have to flame between every return to the plate to get all the cells off the 1/4. Make sure you flame the first time. Do try to use aseptic technique because you don't want to introduce a bunch of contaminants. If you do in this exercise, you'll see them later. We'd rather not. Don't mix up the colors. Again, blue is PUC19, orange is PGEM, and green is PGLOW. The plates are labeled with the E. coli types, but the plasmid type is much more important. Make sure you put the right plasmid in the right color. I'll put this on the board also so we don't mix them up. So we're going to follow the instructions that are in the lab manual. I'm going to put them on the board and we're going to go through it step by step. Again, use aseptic technique throughout. It's a good idea to wear your lab coats because you are playing with Escherichia coli. And this extraction is worth 5 points if you do it right. If you don't do it right, you won't get any plasmids. And then you don't get 5 points.
Oh, good thing I got it. It's
So,
really
the
important.
first thing that you have to do is come up and get the tubes of set buffer. So each group of 3 people needs 1 blue, 1 green, 1 orange.
Is it paused?
And I'll get your E. coli. The next step, to add 350 microliters of lytic mix to your colored tubes. That's a blue pipette. Make sure that you have a blue pipette and 1 box of tips. And make sure your pipette is set for 350. And then make sure you use the plunger properly. Measurement is very critical to this exercise. Blue pipette tips are in the box that has a blue base. Well, some of them don't. Sorry. They're just blue pipettes. Remember, don't leave the boxes open. They're blue. They're fine. They're beautiful. Some of the boxes are purple.
I know. The line was too long. I'm waiting.
Remember, push the button in to reset your pipette. Do not turn that thing without holding the button in.
And we put that in each of these?
You're going to put lytic mix into each one of your tubes. Then you're supposed to invert several times. Set them on ice.
Invert and then put on it.
What is it? What's in there?
I don't know.
Wait, I'll go find
She said invert after you like put it in. Invert and then put on it.
So just like
put it in micro. Yeah. Okay.
oh yeah, just mix it.
Oh. Where's our mix? Is it in that ice bucket?
No?
No, not.
Oh, you got it? Okay.
Oh, I was wondering.
Okay, here, you hold these open, and then I'll hold this open.
Is this still playing?
No. Oh wait, is it? Yeah.
No, yes.
And then put them on ice, and I want to talk to you about the semester project.
3 minutes.
Yeah.
Not you. Using a blue pipette, and you pull liquid up into the tip, and there's a gap in the liquid, it isn't right. Put it out and take it up again. Take it up slowly so you don't get that gap, that bubble. Because a bubble isn't liquid, and those things don't measure properly if you don't have the volume of liquid in a solid mass. That is, it's not solid, it's liquid. No bubble.
Wake up.
You said wake up.
People in the front row, please sit down so people behind can see.
Yes.
Semester project. This is a 70-point assignment, and basically you have to do a report, but it's in manuscript format.
I don't know what that means.
It's on the website. Rather specific about how this thing is going to be written. There's an introduction,
Momentary.
which tells why you're doing this. And the introduction has to have some background information because you have to have a hypothesis. That is what you expect to find. You're going to have to do some research. And it can't just be look on the internet. You can't just go to the internet and pick up any little scrap of information about, say you want to look at organisms from water. If you just go to Google and say bacteria in water, you'll get hits. But in order to be incorporated into this, because it's manuscript format, you have to have an author and a date for your references. So you have to have this. For your citations in the text, you always write it this way, author, date. They have to be in there. Your introduction should have at least 3 or 4. I think there's specifications on the website. There's specifications about how many you have to have. It's in the lab manual as well. Tells you you can have so many from the internet, but they can't all be. A really good source is NCBI, the National Center for Biotechnology Information. They have something like 80,000 free full-text articles. But anyway. You can also look at, if you look something up on Wikipedia, you will notice that Wikipedia has a bibliography at the end, and they can tell you specific places to get information. Don't just take the information off of Wiki, go look at their sources. You want specific references where you can get an author and a date. In this introduction, you also have to include your hypothesis. But you don't state that it's your hypothesis. You just state what you expect to find, because that's what your hypothesis is. How can you expect anything? You have to do research. Otherwise, you don't know what you expect to find. Maybe you're doing organisms associated with air. Well, what do you expect to have in air? Where could the organisms in air come from? They could come off of animals, they could come off of people, they can come from dirt, they could come from water, maybe they came from Safeway, I don't know. But think about where they might have come from and then do some research about the kinds of organisms you can expect to find. Your hypothesis, when I'm saying what you expect to find, probably a number of organisms, specific genus names of organisms you expect to find. Then you have your materials methods. This is written in 3 sections, where your first section is your collection, sample collection, and isolation, and then morphology. Well, I shouldn't put that on there. Because you don't put any data in this section, so your sample collection, your isolation, and then how you studied morphology. So steps to determine morphology. For the semester project, do only 2 stains, gram stain and indirect. Do not do endospore stains, do not do acid-fast stains, don't do capsule stains. You only need 2 stains, gram and indirect, that's it. As far as how do you describe steps to determine colony morphology, well, you're doing observation of colonies. So you're looking at colonies on plates. The second section has to do with DNA analysis. This is somewhat difficult because, for the most part, you guys aren't doing the hands-on handling of the DNA, but it's the same thing that we did in lab. So for the DNA analysis, it starts with chromosome extraction. And then PCR. This is in the lab manual. So the the exercise that has to do with application of the PCR and bacterial identification, that's where you get that information. How did we extract chromosomal DNA? It's exactly what you did in lab for physiological unknown 2. So it's the same procedure, you guys just aren't actually doing it yourself. So the chromosomal DNA extraction happens, then the PCR happens, then we have to do gel electrophoresis. This is to see if the PCR worked. What is the PCR for? Remember? You're amplifying DNA in vitro, which means you should get enough DNA that it's clearly visible in a gel. So I have to run the gels to see if the PCR worked. There's specifics about how that's done. Again, in the lab manual, we will do an exercise on gel electrophoresis, and you get to do gel electrophoresis with your physiological unknown 2. It's the same thing that we did, that I do for the semester project. If there's DNA present, the DNA must be purified. This DNA purification business is very much like following a cookbook. So we have these little boxes. They come from a company called QIAGEN, spelled Q-I-A. There's no U after the Q. Anyway, QIAGEN has a little booklet, and the booklet tells you what to do. So you just extract chromosomal DNA following the procedure that is in the box, QIAGEN box. You don't have to put down all the steps of the DNA purification. You guys don't actually get to do it. But anybody can do it if they just buy this box. So this is a QIAquick PCR purification kit. We're actually using a QIAquick gel purification because we're running the samples into Agarose before we do the purification. Once the DNA is purified, that DNA is taken to UC Davis. And Davis does sequencing. This is what we should have been doing on Tuesday. Last Tuesday, we would have talked about nucleotide sequencing and how this works and what electropherograms are. We're going to do this lab. I'm just not sure how or when. We can't not do it. We have to talk about the sequencing and look at the results of the PCR. So the DNA goes to Davis, they do the sequencing, they send us AB1 files. These are .ab1. They're on the website for your physiological unknown 2 because we're going to use the physiological unknown 2 exercise as a way to show you how to work with the electropherograms. We typically have 3 primers that we're running. For the semester projects, I'm not sure how many sequencing primers we'll use, at least 2 per sample. So you're going to look at these AB1 files, you're going to look at electropherograms. And what you have to do is put a series of electropherograms together so that you get a contiguous sequence. What we're trying to get for the DNA is 16S ribosomal RNA gene. It's coming out of cells, so a gene is DNA. So you could say, well, it's 16S ribosomal DNA. It is, it just causes people confusion. Is it RNA? Is it DNA? It's the ribosomal RNA gene, it's DNA. Anyway, then we're going to be using an algorithm called BLAST. This is associated with the National Center for Biotechnology Information. Which is called NCBI. Anyway, NCBI has this BLAST algorithm, and you compare your nucleotide sequence to whatever is in the database, the gene bank, And that's how you find out what your semester project organism actually is. This is also how you find out what your physiological unknown 2 is. So you're gonna have to do this, like I said. I'll put something up, and I'll see if people can work on it at home. If you can't, then we'll figure out a way to get it done in here, because you have to do it. You have to learn how to do this. Anyway, the third section, so this is section 1, section 2. If you do a third section, it's enzymatic tests. In the other lab sections, a great deal of emphasis is put on these enzymatic tests. You're gonna do all these enzymatic tests and you're gonna identify. I've taught this class for more than 40 years, and what I came to the conclusion was, that doesn't work. You can get completely lost with morphology and enzymatic tests and you have no idea what you've got. But if you sequence, you're gonna be in the right family and probably in the right genus. And then you can do some enzymatic testing to verify, and that's what we're going to do. We're not going to put a whole bunch of emphasis on doing a bunch of enzymatic tests. So in the lab manual, you have this big long list of tests that you can do. Please don't go there. It's a waste of media. If you have a Pseudomonas, you run a whole bunch of enzymatic tests for fermentation of 40 million different carbohydrates. Pseudomonas don't ferment. It's worthless. And we don't have single carbon source media, so don't do it. Please don't do it. I'll let you know what kind of enzymatic tests you might get to run after we get this DNA data back. That's why it's so important we get this done. So I'm hoping by next week we'll have the DNA will go to Davis. I'm hoping to get it down there on Monday. By Tuesday we might have data back. If we don't, we'll certainly have it by Thursday. Yeah, they're fast. They say they have a 3-day turnaround, but it's faster than that. So this bothers me a bit because there's so much stuff happening here that you don't actually do. But sample collection and isolation, you guys got to do that. As far as the chromosome extraction and PCR, you do that for physiological unknown 2, and it's the same steps. The gel electrophoresis, you're going to get to do. We haven't done that yet, but you're going to. The only thing you don't actually do is the DNA purification, and then you have to deal with the sequences. You're going to have to do this editing and putting your contiguous sequence together, and you get to run the blast. So you will do this. We just haven't done it yet. So after that, there's a section on results. The results, I would like your results to be in the same 3 component. So you have results of your colony morphology, your cell morphology. You do a gram stain on a gram positive, gram negative, what's their size, what's their shape, etc. The results that you get from the blast goes on there, and then the results from enzymatic testing, if you do any. And then there's a conclusion or a discussion section at the end. But exactly how this thing is written up is pretty specific. It all has to be third person past tense. You can't use I, my, mine, even we. It all has to be as though it was done by somebody else, or somebody that we don't know exactly who it was. Somebody did it, and they did it months ago. It was done. It was accomplished. Don't use that word either. Done's a dead word. This project was accomplished by. It all has to be third person. It all has to be past tense. And that's because in reality, research takes time. It can take years to get things done. So it isn't, I did this today. It's never present. It's always past tense. Okay, anyway, is it time's up? I hear this.
Yes, sorry, that was moving away.
No, that's fine. Time's up, so we have to go on to our next step. Okay, so what you're going to do at this next step is we're going to remove all the tubes from the ice, and we're going to put them into centrifuges, and we're going to centrifuge them. They're going to centrifuge for 10 minutes. So is everybody's sodium acetate ice bath finished?
We have like 3 minutes.
Some of you are not quite finished. Those of you that are finished early, leave yours on ice. It's okay for them to be on ice longer. We only have 3 centrifuges back here, so when we load the centrifuges, we want to get, each one can run 12 max. I'd like at least 8 tubes per centrifuge. Otherwise, we run out of space, especially because it's such a long run. So, if you have your time's up for your sodium acetate, just let them sit on ice. I'm going to put material out here. The next thing that you're supposed to do is pick up a clean, sterile microfuge tube that has a sample that is the same color. So where did we get to on this? We've done this. You want to get tubes of the same color. When you close these tubes, put your thumb on the back of the cap. Be really careful you don't touch the inside of the cap or the inside of the mouth. These samples are ultimately going to end up in the same colored tube. So if you have an orange sample, it goes in an orange tube. If you have a blue, it goes in blue. If you have, anyway, right now you're just picking up the tube. And then hopefully then everybody will be at the end of the 20 minutes and we'll load the centrifuge up. Be careful about, don't tear the aluminum foil, please. Be careful about these tubes. Try to use aseptic technique as much as you can. Heat is the liquid that's above that. The cellular debris is cell wall material, chromosomal DNA, and whatever else was in the cell, all tangled up with the precipitated chemicals that are in there. So it's going to be this white blob at the bottom. What you're going to do is, after these come out of the centrifuge, very carefully pour your Supernate, the liquid, into your new clean sterile tube. So
that's why we have those two.
this step on the board.
Here's my little thing so I can spit it over with my hand. Wow, freaking hot
Why
glass.
would you do that?
Heat fix.
Why would you do it with your hand? The thing's right there.
Oh, I didn't see it. Okay, what do we do next?
You put it on there? I don't think you put it on.
Rather than pouring the liquid,
Okay, we're
then
done.
you use a pipette.
Fix it.
You're going to get plenty of liquid if you just pour it across. But if you pour it carefully into the new clean tube, then look in there, and if you see material that kind of looks like meringue, kind of white, gooey looking material, reach in with a sterile loop and take it out. That's protein. We don't want that hanging in there. It should have gone to the bottom. So you're going to pour the liquid into the clean sterile tube and then close it. And you got to label that top. I forgot about that part. Sorry. Well, you have to do this, remove any floating white goo first. Anyway, once you've done this, you close your tube, you label the top. Then we're going to put some RNAs in there, which I haven't passed out yet. Oh. Well. So when those guys come out of the centrifuge, I'm going to set up a flask up here at the front. The flask will have disinfectant in it. After you've carefully poured your liquid into your new clean sterile tube, you're going to discard the cellular debris, which is this colored white goo, by dropping it into the flask with the disinfectant. Leave it open. That's just going to be thrown away. What you're keeping is the liquid up top. That's where the plasmids are. Let me get that flask set up.
for use, but that was my